We need to contrast skeletal and cardiac muscle because there are quite a few differences. When we learned about skeletal muscle, we learned that each individual muscle fiber needed to be stimulated by a motor neuron if we wanted it to contract. But that's not how it is with cardiac muscle cells. We have individual cells called autorhythmic cells, and these autorhythmic cells, they're not contractile cells, but they're little cells that are self-excitable. In other words, all of a sudden they create their own action potentials, and then they're going to spread those action potentials to other cardiac cells, and those other cardiac cells are actually the cells that go on to contract. So there is no need for outside motor neuron innervation. So that's very different than the skeletal muscles that we learned about last semester. Big difference. And in skeletal muscles, we learned about motor units, where we had a group of skeletal muscle cells that had a motor neuron that came into them that innervated them. And so when it came to skeletal muscles, we could have varying levels of muscle contractions. We could have weak muscle contractions or strong muscle contractions, and it would depend on how many motor units were recruited as to how strong the muscle contraction was. So the whole muscle didn't have to contract, but that's not how it is with the cardiac muscle. With the cardiac muscle, the whole muscle has to contract, because if one cell contracts, the whole muscle contracts, because each cell is connected to all the other cells by gap junctions. So that means that the cardiac muscle acts as a functional syncytium. The whole heart acts as one. That's what a functional syncytium is. So it's an important concept. Now, in skeletal muscle, we have what's called a short absolute refractory period. It only lasts 1 to 2 milliseconds compared to how long a contraction is, which could be anywhere in some of our eye muscles from 15 milliseconds to our other muscles. The contraction will last 100 milliseconds. So we have a short absolute refractory period compared to a long contraction. In cardiac muscle, we have a long absolute refractory period at up to 250 milliseconds, and that absolute refractory period lasts as long as the actual contraction. The contraction also lasts 250 milliseconds. And the reason we have this is it prevents tetany. Tetany is a state of sustained muscle contraction. We do not want our heart to go into a sustained muscle contraction. That would be horrible, because then our heart wouldn't sufficiently pump blood out to the rest of the body. So that would be horrible. And recall that absolute refractory period is a time that a muscle cannot possibly be stimulated no matter how strong the stimulus is. So we'll define absolute refractory period again here in a little bit and tell you why an absolute refractory period occurs when we get to our graphs. So here's what cardiac muscle looks like underneath a microscope. Remember that it's striated and it has the intercalated disc, and usually there's just one nucleus to each little cell, and there's a lot of connective tissue between the cells, and this connective tissue acts like an origin and an insertion to give these cells something to push and pull against when they contract. Here's our intercalated disc between the cells. These are where the cells connect to one another, and they pick up the stain very well. That's why we see them. It's just a region full of lots of stuff, and that stuff would be like desmosomes. Remember that desmosomes are adhering junctions. These cells are going to, when this cell contracts and this cell contract, they're going to pull apart from one another. And when they pull apart from one another, we don't want them to rip apart, so those desmosomes make sure that they don't rip apart. They're adhering junctions. Gap junctions are also found in these intercalated regions, and the gap junctions allow ions to flow from this cell into this cell, and that's for communication purposes. These gap junctions are the reason that the cardiac muscle acts as a functional syncytium, acts as one big unit. Another thing I want you to notice in this picture is you see all the sarcoplasmic reticulum. This sarcoplasmic reticulum is the organelle that stores our calcium, so we don't have just calcium floating around in our sarcoplasm, because calcium is the final trigger for muscle contraction. Remember that. because this muscle will contract when calcium comes out of the sarcoplasmic reticulum. Go back to your skeletal muscle chapter and remember everything that happens with troponin and tropomyosin and our Z disc and the moving of the thin filaments over the thick filaments, and look over your I bands and your A bands and everything that goes on. It'd be exciting. Read over your old discussion questions from your last test. Wouldn't that be exciting for you to review? I know you're excited just thinking about it. Here's your one little nucleus inside this cell, and here's your mitochondria. Look at all the mitochondria shoved in there. And that's because your cardiac muscle has lots of mitochondria because it needs it for energy. And we have more mitochondria in cardiac cells than we do in actual skeletal muscle cells because your cardiac muscles are constantly contracting. Contracting. Since, your cardiac cells were formed in you in a fetus and they'll beat until you die. So we have more mitochondria in these cells than in skeletal muscle cells. And I think that's all I want to point out here. I'm looking around to see if there's anything else, and I think that's it. I know you recognize this graph. This is an action potential. This is an action potential that would occur across the sarcolemma of a skeletal muscle cell or across a neuron, across the plasma membrane of a neuron. So if we apply a stimulus, here we have the negative 70 millivolts here, and once we have depolarization up to threshold, then the sodium gates are going to open, more sodium gates are going to open, and we're automatically going to have this action potential. It's all or nothing. Sodium gates are going to open, sodium influx, sodium's going to rush into the cell, and then the sodium gates are going to close. Remember, these are going to be voltage regulated fast sodium gates, so they're all going to open up. And then the sodium gates are going to close, and potassium gates are going to open, and we're going to have repolarization. Potassium's going to leave the cell, and those potassium gates, the reason they opened up second is because they're just slower to open. The same stimulus that caused the sodium gates to open also caused the potassium gates to open. They just opened up slower. And so the sodium gates had closed, potassium gates opened, potassium is coming in, and then those potassium gates just closed a little too slow, and so we have this after hyperpolarization or this undershoot, and then we go back to normal, back to resting membrane potential. So this is a normal action potential. And here's our absolute refractory period. It's a time where we can't possibly stimulate the cell to have another action potential. This absolute refractory period ensures that every single action potential is going to be a separate event, basically. And you cannot possibly stimulate this cell during this time, this absolute refractory period. During the relative refractory period, you could stimulate the cell, but it would take a stronger stimulus because we've hyperpolarized the membrane. But we'll just ignore that for right now. And during the absolute refractory period, you can't stimulate the cell because all of your sodium gates are open here, so you can't open anymore anyway. So this is a normal action potential. This is not what happens in your heart. This would occur in neurons, this would occur in skeletal muscle cells. But I said that in cardiac cells, the absolute refractory period doesn't last just 1 to 2 milliseconds, it lasts like 250 milliseconds. So this line would keep on going times, a couple, times 200. It would be, very long. So here's what an action potential looks like in cardiac cells. It looks more like this. There's this big plateau, and look at the extension here, time in milliseconds. Goes out to 300. This time in milliseconds was, the whole action potential lasted 3 milliseconds. Now our action potential is lasting, up to 300 milliseconds. So this is a crazy looking action potential. What happens in cardiac cells, and I'm sorry that these aren't labeled here, so pay attention. In cardiac cells, we get a stimulus, and sodium gates open, and sodium rushes in, just like in our neurons and our skeletal muscle cells. So we do get our depolarization. We're going to be positive here, but the sodium gates are going to close, And then other gates are going to open to create this plateau here, to keep us depolarized for a longer period of time, so we'll have a long absolute refractory period. See in gray here, in this dark gray region, this is our absolute refractory period. And what elongates this absolute refractory period is the fact that we're staying positive because we've opened up a second set of gates, and those are our calcium gates. And so our calcium gates open up, and they're slower than the sodium gates. That's why they open up second. And when the calcium gates open, calcium comes in, and it keeps us positive for a longer amount of time. And then our potassium gates open, and when potassium gates open, we repolarize the cell and we go back to negative again. And it looks like this. So this plateau is created in our action potential by a second set of gates opening, the calcium gates. And that's just not happened to us before, in anatomy and physiology. So in these cardiac cells that are contractile, they just have a different type of action potential altogether. So in this graph over here, it's not a graph that I would have you understand. Wait till my bird stops singing. It's not a graph that I would have you label on a test or anything, but it just shows you that right here, the sodium gates are opening because the sodium permeability of the cell is really high here. And then the sodium gates close. This is when sodium's coming in. And then next, potassium gates open. Calcium gates open. And then the calcium gates close. Next, potassium gates open. So it's just showing you the order. Sodium gates open first, then calcium gates open, then potassium gates open. So in summary, if we compare and contrast these action potentials in skeletal muscle cells, the first thing that happens during depolarization is that fast sodium gates open, causing depolarization, and then during repolarization, the potassium gates open. In cardiac muscle, depolarization is caused by two things. Those fast voltage-regulated sodium gates open, just like over here, plus then we have slow voltage-regulated calcium gates open during this region that cause the plateau. And then during repolarization, the same exact things are occurring. Potassium gates are opening. So really, you just have one more step to learn. The same exact thing is occurring on this side, fast voltage-regulated sodium gates, fast voltage-regulated sodium gates. On this side, during repolarization, potassium gates open, potassium gates open. You just have one extra step to learn right here. Slow voltage-regulated calcium gates open, causing a plateau. And this elongates the absolute refractory period to make sure that our heart doesn't go into tetany, that our heart doesn't lock up, and make sure that heart contractions, your heart's always going to be beating, and your heart doesn't lock up, your heart doesn't go into tetany. Sound like I'm stuttering. Okay, now pacemaker potentials, in comparison, pacemaker potentials are done by autorhythmic cells. Autorhythmic cells are those cells that are not contractile in the heart. So we've got a totally different type of action potential that's about to occur, but they're not really action potentials. Let me show you the graph before we talk about this. So here's an action potential that's going to occur in an autorhythmic cell. In this case, here's the action potential, and here's the second one. We have, this is totally different than what you just learned about. In this case, we're going to have sodium gates that open up. But these sodium gates are called slow voltage regulated sodium gates because they're very slow to open. And basically you can think of sodium just very slowly leaking into the cell. And when the sodium slowly leaks into the cell, when enough sodium gets in for that cell to reach threshold, all of a sudden calcium gates pop open, and these calcium gates are called fast voltage regulated calcium gates because they all pop open at once, and calcium rushes into the cell, and that causes a depolarization of the cell by calcium. something you've never seen before. And then those calcium gates close, and then we get the normal repolarization by potassium. Then those slow regulated sodium gates open. Sodium starts to sneak into the cell. When enough gets in to reach threshold, those fast voltage regulated calcium gates open. Bam, depolarization again. And then we go back to repolarization. So what happens here in these autorhythmic cells is not a normal action potential at all. We call these pacemaker potentials. What these autorhythmic cells are doing is they're firing off these pacemaker potentials, or these are actually, this region right here is the pacemaker potential. This is the action potential, but I'm going to call the whole thing a pacemaker potential just to make it easier on me. These autorhythmic cells are firing off these pacemaker potentials, and they're exciting all the cells around them. They're firing off these pacemaker potentials. They're irritable cells, and they're just unlike all the other cells of your heart. They're not going to contract, these cells. They're just those excitable cells that are causing all the other cells in your heart to contract. So these potentials occur in the autorhythmic cells, which are non-contractile, and they're just way different. So here's the steps again. You have slow voltage regulated sodium gates. They open to allow sodium to leak in. And then once you get enough sodium to leak in to reach threshold, you have fast voltage regulated calcium gates to open. So calcium rushes in, causing depolarization. And then voltage regulated potassium gates open, repolarizing the autorhythmic cell. Then the wave of electricity that was just created by those autorhythmic cells is passed along to excitable contractile cells of the heart, and that causes the heart to contract. This wave of electricity causes those cells, those contractile cells, to create action potentials like this along their membranes. And then these action potentials along their membranes leads those cells, the cardiac muscle cells, to contract. It's all very fun learning all this good stuff. These pacemaker potentials, are created in autorhythmic cells, and all these autorhythmic cells are found in these regions of the heart. The SA node, the AV node, the AV bundle, the right and left bundle branches, and their Purkinje fibers. SA stands for sinoatrial node. The AV stands for atrioventricular node, and this is the atrioventricular bundle. And that's what we're going to talk about next in the next lecture.